Good morning, professors. I'm Dr.G. VARATHARAJU, Assistant Professor, Department of Botany, Sri Kaliswari College, Sivakasi. First of all, I would like to thank the organizing secretary for giving me the opportunity to share my knowledge with you. I'm going to share the information on bioinformatics, tools and applications. In the field of bioinformatics, the important one is biological databases. So in this lecture video, we will discuss about biological databases. Bioinformatics is very important field for all kind of biological research. Bioinformatics is a hybrid field which combine biological data with computational science, computer science. The outcome is bioinformatics. We'll see the bioinformatics in different headings: definition, biological databases, genomic research, proteomic research, and bioinformatics, how it is applied in different fields. So finally, the outcome of bioinformatics, particularly the biological. The field of science in which biology, computer science, and information technology merge into single discipline, which is called bioinformatics. In laboratory, biologists, what they will do, they will collect molecular data such as DNA and protein sequences, gene expression data using different technology like microarray technology and so on. The computer scientists, they will develop different tools, softwares, algorithms to store and analyze the different types of data. So what we are doing is, we are combining the molecular biology data from biologists and the algorithms developed by computer science scientists. These two are merged together and will develop a new field, that is bioinformatics. So that is why I clearly mentioned that the bioinformatics is a hybrid field where biological data are merged with computational algorithm. It is called as bioinformatics. Molecular bioinformatics. Molecular bioinformatics involves the use of computational tools to discover new information in complex data set. That is one-dimensional information of DNA through two-dimensional information of RNA and three-dimensional information of proteins. So finally, four-dimensional information evolving living systems. So the one-dimensional data, two-dimensional data, three-dimensional data, four-dimensional data are interrelated with each other. If we have any one of data, will derive the another kind of data with the help of different tools available in both online and offline. Bioinformatics is multidisciplinary field which includes molecular biology, genomics, mathematics, statistics, numerical analysis, algorithms, data management, image analysis, evolution, biophysics, biochemistry, genetics, and etc. Well, biological databases. Biological databases are important in bioinformatics. Biological database is nothing but it's a database used to store biological information. There are different types of biological databases are available online. From the biological databases, what we are expect, that is sequence. So we are expecting the biological database to provide sequence information, functional information, structural information related to bibliography. The bioinformatics, the moment we are accessing biological database, the biological database must provide specified sequence. or expected sequence. At the same time, the functional information related to the particular biological data, and also it should give structural information. So the sequence, that is primary duty of biological databases. The biological databases stores the sequence information, and whenever we need the sequence information, we will retrieve the information from the database. And also it must provide the functional feature of the biological data and the structural information. And if you take any biological database, the database must well structured and indexed. This is very important. Once it is indexed, the accessing of biological data will be very simple and useful. And if when biological database is well structured, it is very easy for the users. Without any difficulty, the users will access the specified data from the biological databases. So that is why it is considered as important criteria for biological databases. The biological databases must be well structured and indexed, and also it must be cross-referenced. Whatever the data present in the biological databases, it must be well referred, or it must be interconnected with another databases. That is very important. The interconnection is because of the validation of the biological data present in the database. Another foremost important future for biological databases is, once the biological databases is created and it is updated, I mean it is stored with biological data, it must be periodically updated. Then and way the scientist can retrieve information currently. Otherwise, the old data will not be useful. So, the biological databases must be updated frequently. That is very important. And finally, it is another important parameter for all the biological databases. The biological databases may have a huge number of biological data, but it must be provided with tools for the analysis of biological data present in the database, and also it should be provided with tools for visualization of the data. That is very important. So these are the expectation of biological databases. So we all know that the cell is a house to maintain all the cellular components. The cell, if you take any cell, the cell is very well protected with a cell membrane. Inside the cell membrane, we can see protoplasm. The protoplasm contains nucleus and the cytoplasmic organelle. We are here we are focusing on the nucleus. The nucleus is one of the important source to provide genetic material in the form of DNA or the chromosome. DNA, deoxyribonucleic acid, it is very important to retrieve biological data. There are two major parts of DNA. One is introns, another one is exons. The introns are intervening sequence, that is otherwise called as non-coding sequence. That is, exons are expressive sequence, it is coding sequence. There are simply the exons are called as genes. So genes are very important, which hold the entire the genetic property, and it is very important to maintain the internal and morphological future of future of an organism. So genetic material, particularly the DNA, is very important and frequently used for biological people, particularly working in bioinformatics, because the genomic research, the people those who are doing genomic research, they will focusing on DNA, or the scientists who are working in proteomics, again they need DNA sequence, because the protein structural information or the functional informations are retrieved from the gene sequence. The gene sequence, once we have gene sequence, the gene sequence can be converted into amino acid sequence. Once the amino acid sequence is derived or obtained from the amino acid sequence, it can be converted into structure with the help of different offline and online tools. So that is why I am here focusing on the DNA. DNA has many important property for bioinformatics scientists. So this slide shows the formation how the chromosomes are formed from the DNA. The DNA is a thread-like structure. The thread-like structure are compactly winding on the surface of the histone protein. There are four different types of histone protein. The compact nature of DNA associated with the histone protein is called as chromosome. When the chromosome releases histone protein, it forms a linear thread, that is DNA. When linear form of DNA combines with the histone protein, it will form chromosome. It is happening in the cell cycle. So when the cell cycle goes with M phase, we can see chromosome. When the cell cycle goes with the interphase, we can see DNA. And it is continuous process. Alternatively, DNA binds with the histone protein, it will form chromosome. The chromosome releases histone protein and DNA we can see. So this is the cyclic process happening during cell cycle. Okay, the biological databases. In the worldwide, there are three major biological databases are available. It is International Nucleotide Database Collaboration. The fourth and most important database is GenBank. GenBank is maintained in NCBI. And another database is EMBL. EMBL stands for European Molecular Biology Laboratory. This database also stores nucleotide information, and the database has many tools for the visualization and analysis of biological data, and also the database is frequently updated. And the third important database is DDBJ. DDBJ stands for DNA Data Bank of Japan. These three, that DDBJ, GenBank, EMBL, these three databases are interconnected with each other. Because of this interconnection, the data can be shared by these three databases. For example, one scientist are working in the laboratory and they successfully sequenced a specific gene, and immediately the scientist updated the sequence data in the GenBank. So now the data is successfully updated in the GenBank. The data updated in the GenBank can be retrieved from the EMBL database or DDBJ database because of the interconnection. Wherever you are updating the data can be accessed by three different databases. So it is sharing by three different databases: GenBank, EMBL, DDBJ. So that is why these three databases are generally considered as most important databases, and these three databases are frequently updated. Okay, and the data, for to store information in the database, we can work with molecular biology. We can identify the gene with the help of PCR. We can amplify the gene. After amplification, we can send the gene product to sequencing. We'll get sequence. Once we received the sequence can be analyzed and updated in the database. This is one method of getting information. For all the time, this is not possible. When it is so, we must know what are all the ways to get information to store or update in the biological databases. There are different sources of getting information. One is sequence, another one is when we have structural information. From the structural information we can derive the sequence information, or we'll go through the different literature from the reputed journal. From the literature we can get information, or we can get information from the domain or the networks is another important source of getting information. Once we have received information from the any one of the source, such as sequence, structure, literature, domain, or network. So if you have any source, from these source we will get new information. Once the informations are derived, it is a knowledge for the particular portion. The knowledge can be updated in the database. Once it is updated in the database, the knowledge is common for all the people, those who are working in the particular area. So in this slide, we are mainly focusing on the different source of getting information. Here, we'll discuss about the different tools. There are plenty of tools are available both online and offline. Whatever the tools are available, the tools are regularly used in three different areas. One is molecular sequence analysis, second one is molecular structural analysis, third one is molecular functional analysis. For example, molecular sequence analysis, you can use online tool SS FFAS, it is online tool, or we can use Bioedit. Bioedit is one of the most important and frequently used offline tool. It is freely available. And structural analysis. For structural analysis also we are having plenty of tools. The best tool for structural analysis is PyMol. And the functional analysis. Functional analysis also we are having tools. So what we need to have is, we need to have knowledge on the tools, how it can be effectively used for the different purpose. So if we have knowledge on the tool, then we can do wonders. Okay, we'll see one by one. Sequence analysis. Sequence analysis is nothing but sequence alignment. For aligning the sequence, we need a minimum of 2 sequence or many number of sequence. Where we will get sequence? That is very big question. Without any doubt, the databases are the source for collecting sequence. So we will collect sequence from the database. There are different types of databases are available. Already I have told you. Generally, the databases are classified into two major categories. One is sequence database, another one is structural database. So from the sequence database, sequence can be retrieved, and the motif and pattern discovery can be identified from the sequence, or we can identify the gene or promoters present in the sequence, or we can reconstruction of evolutionary relationship. We have many number of sequence, we can identify the relationship among the sequences or among the organisms. So these are the application of and the structural analysis. Proteins and the nucleic acid structure can be analyzed. If we need, we can compare two different proteins or two different nucleic acid, or we can classify the proteins, or we can predict the protein when we have sequence of the molecule. For analysis, comparison, classification, prediction, there are specific kind of databases are available. Not only the database, and also we have plenty of databases. For protein related information, the best database is Protein Data Bank. Protein Data Bank is a popular and frequently used database for protein related information. So there you can analyze the protein, can compare the protein. For classification of the protein, we can use scope or cat. Cat and the scope, these are the two important databases available for protein for classification and the prediction. There are too many tools are available to predict the 3D structure of proteins. Functional analysis. So from the gene, what we can do? We can do gene expression profiling. Whether the particular gene or whether the particular sequence has the expression or not. So this can be easily identified by bioinformatics tools. Protein-protein interaction prediction, whether the particular protein will interact with another protein or not. So this can be identified by the tools, softwares. Protein subcellular localization prediction, metabolic pathway reconstruction, and so on. So plenty of work can be done with the help of bioinformatics. For whatever the work we are trying to perform, for all the things we need sequence. Once again, I am telling you the source for retrieving the sequence is biological database. So that is why I am giving more important for biological databases. So this already we have seen. Okay, the databases. So this slide clearly shows the types of data and the types of databases. There are three different types of data: primary data, secondary data, and tertiary data. The primary data is nothing but sequence, like DNA sequence or amino acid sequence. Secondary data is nothing but motif, regular expression, blocks, profile, fingerprints. These are all the example for secondary data. The secondary data stored in the secondary databases. The secondary databases contains secondary protein structure, that is alpha helix, beta strand, and so on. Tertiary data is nothing but atomic coordinates. Atomic coordinations are seen in the tertiary data. The tertiary data stored in the tertiary databases. The best example for a tertiary database is PDB, Protein Data Bank. There you can collect domains, folding, unit, or entire protein complex. The scientists are purifying the proteins and they have constructed 3D structure with the help of NMR or X-ray crystallography. Once it is constructed the structure using this technology, that is NMR or X-ray crystallography, immediately the scientists are storing their structural information in the protein data bank. So without any difficulty, we can collect 3D information of the protein from the protein data bank. The specialty of the protein data bank is we can get 3D structural information, 2D structural information, and 1D structural information, that is sequence information. And also the protein data bank provide lot of tools for different analysis and prediction. So that is the specialty of the protein data bank. Databases are classified into different categories. First, the database are classified into nucleic acid database and protein database. The example for nucleic acid database is EMBL, European Molecular Biology Laboratory. GenBank. The GenBank is available in the NCBI. DDBJ. DDBJ stands for DNA Data Bank of Japan. So these are the popular databases available for nucleic acid. And The databases for proteins, PIR. PIR stands for Protein Information Resource. PIR database, PDB database, Protein Data Bank database, and the SysProt database. SysProt means the protein database developed by Switzerland. So it is called as SysProt. SysProt. TREMBL. TREMBL is sister concern of the EMBL database. So the specialty of the TREMBL is the database stores translated information. So that is why it is called as TR. This slide shows international nucleotide data bank. So already we have discussed. So in this slide, how the international how the databases are operated. The databases, already I have told you that the three databases such as GenBank, EMBL, DDBJ are interconnected with each other. These three databases are governed by International Advisory Meeting Collaborate, International Advisory Committee. So this committee join together, collaborate the information and share the information, and this committee is responsible for the maintenance of the database. So this already we have seen. So this slide, another important slide for biological databases. Already I have told you that the databases are two different types. One is nucleic acid database, another one is protein database. If we have nucleic acid, from the nucleic acid we can convert protein. Once the protein is converted from the nucleic acid, immediately we can update the data in the protein database. Or sometimes you may have protein with you, amino acid sequence with you. If you want to see the amino acid coding DNA sequence, so we have tools. With the help of tools, translate tool. The translate tool is used to translate DNA sequence into protein. Reverse translate tool is used to translate amino acid sequence to DNA sequence. We can convert DNA to amino acid and amino acid to DNA. It is very important slide for learning biological databases. And for biological databases, the central dogma is very important. As we all know that DNA is genetic instruction or gene. DNA synthesize the RNA, and from the RNA we can see the formation of the proteins. DNA to RNA is called as transcription, and the RNA to protein is called as translation. Altogether called as protein synthesis. It is very important because the two different data, amino acid sequence is entirely different from nucleic acid sequence, but though it has entirely different from each other, we can convert nucleic acid sequence into protein sequence, amino acid sequence, or from the amino acid sequence to protein sequence. So this is possible by the development of softwares by computer scientists. We know, as biologists, we know the biological information and the nature of biological data, but we don't know the algorithm to develop. The computer science people are developing algorithm to satisfy our need, and we are using the algorithms for our convenience. So finally the field developed bioinformatics. So this is because of the complete knowledge on the DNA and the protein. So that is why I am repeatedly telling you about the DNA and the protein. So how it is possible? That is the conversion of nucleotide sequence into amino acid sequence. So this is possible by developing algorithm based on the nucleotides. There are 4 different nucleotides: adenine, guanine, cytosine, and thymine. We know how the nucleotides are base paired with each other. With this information, or based on the association or combination of the nucleotide, we'll develop algorithm to encode the amino acid sequence. Here we'll see where do the 3D structure come from. So already I have clearly mentioned that if you have, or when you are working in genomics lab, gene information can be retrieved, or genome information can be retrieved. Once the gene information is retrieved, the gene sequence can be converted into amino acid sequence. Or if we have facility to work with NMR or X-ray crystallography, these are the 2 methods to construct protein structure, protein 3D structure. Otherwise, we have to convert nucleotide sequence into amino acid sequence. From the amino acid sequence, we can construct 3D structure of the protein with the help of molecular modeling. For anything we can do in bioinformatics, whatever the work we are performing in bioinformatics, the raw material is very, very important. The source for the raw material is biological databases. So huge number of databases are available. The application of bioinformatics or biological database. The application of bioinformatics is molecular modeling. We can do the modeling of the protein. We can design a drug. Once the receptor structure is modeled, or the drug is designed, immediately we can do docking, how the ligand is interact with the receptor. We can use gene identification, gene annotation, gene expression, gene functional prediction, phylogenetic reconstruction, metabolic pathway. So we can perform these many work in different system. We can perform all these method in animal system or plant system or microbial system. So if you have interest in any one of the system, we can apply this method in animal system, plant system, or microbial system. So this is the homepage of NCBI, that is GenBank. GenBank is the premier database for nucleotide. This is the homepage of Protein Data Bank. It is the premier database for protein. For all kind of protein information, we can use Protein Data Bank. And so here we can see 2 options. One is display files and download files. If you need sequence information or structural information, you can download the files from download files. Or if you want to see any more information regarding the protein, we can use 3D view, annotation, experiment, sequence, genome, version options. These options will provide you detailed information of the specific information. For example, if you want to see the 3D structure of the protein, we can go with this tab, 3D view. For some people want to know how the proteins are crystallized, how the proteins are isolated, where the proteins are isolated, what is the source of the protein, and who isolated the protein. For this information, for these questions, the answer will be provided with the experiment. If you click this experiment tab, this will give you all your all answer for your queries. Sequence. If we want to see the sequence of the protein, you can use this tab button, sequence, and so on. So it is user friendly database. You can work with the database. When you are working you will get familiar in the protein data. With this information, I conclude my talk. Thank you, Professor, for your patient listening. Thank you all. These are the reference book. If you are interested, you can refer this book, and these books will provide you plenty of information on the biological database, sequence analysis, and etc. Thank you. Thank you all.